Today's topic will be mainly about some image analysis tools applied for bioimaging, specifically fluorescence microscopy imaging. So a little bit about who we are. So as Wen just introduced, so I am the technical director of the Multiphoton and Analytical Imaging Center at the URMC. We are part of the shared resource labs, so we provide equipment services to all the researchers on campus or in the nearby institutes. We work closely on a daily basis with the biomedical researchers and from all different departments to conduct mainly for micro multiphoton fluorescence imaging. And also we collaborate with biologists to help them do image analysis. And also I will be very glad if I can see more collaborations with other data scientists to work on some of the bioimage analysis problems we have here, because on daily basis we see a lot of different image analysis problems that biologists have. So for today's talk, I will mainly focus on two projects we have been working on in the lab that we saw those difficulties when we conduct imaging using the fluorescence microscopy. So we look for ways that has been fully developed in other fields that are easy and simple to use, and try to apply it for the bioimages. And we want it to be easy to use, but in a robust way. Before I go to the details of those two projects, I first developed this section as an introduction of fluorescence microscopy basics, the applications, and also the limitations as introduction to highlight what kind of limitations we have with the fluorescence microscopy. Some of you may know about fluorescence microscopy. Here is just some basics about the regular fluorescence microscopy. So it is a group of microscopy techniques. So underneath it, there are many different types, but the bare bone of it is very simple and highlighted here. So with any fluorescence microscopy, you need a light source, which is usually a laser for our case, as excitation light source at a certain specific wavelength. And then it will be directed to the sample, which is fluorescently labeled with a certain fluorophore in a specific tissue structure, for example. And that fluorophore can be excited by the laser and then emit at a certain wavelength. And that emission signal will be detected by detectors. And for our case, the most commonly used detectors are photon multiplier tubes, which are a little bit different from the regular camera we use. So here we will need a fluorescently labeled sample first. And also the commonly used fluorescence microscopy techniques such as confocal microscopy and multiphoton microscopy. Sometimes you may hear two-photon microscopy. It's basically very similar things here with the multiphoton microscopy. So the nice thing about multiphoton microscopy is that it can image very deep into the tissue. And comparing to the traditional, what we call one photon microscopy techniques, such as confocal. And just as a reference, usually we can image like until 400, 500 micrometer depth easily. Comparing to confocal, usually it's within 50 micrometer depth. And because of this uniqueness, and the multiphoton microscopy have been very commonly used for thick tissue sections imaging, so people don't need to slice it to make it a slide. And also it's been more commonly used for live animal imaging, because you don't, you can image the animal live and the people can study the tissue under the most natural environment. Are there some questions?
Can I delete these things or no?
Someone has a question? I cannot hear. Hello?
I think it was simply somebody background noise. Background noise. That's it.
Yeah, feel free to chip in if you have any questions.
Okay.
So it has been commonly applied for biomedical research to study tissue structure, cellular dynamics, disease mechanisms, and so on. Here, I would like to show you 3 examples of the images collected under multiphoton microscope. So the first one here is a human lung tissue, and this is a 3D image on the fixed sample, and there are 5 colors labeling. And what we are looking at is from the surface of the tissue to the bottom, and to the very bottom we can see. So it's a loop played of the 3D. And the second example here is a live mouse skin image. And this is a 4D image, which my user collected a 3D image and repetitively and form a time lapse here. And what you can see moving inside the tissue here are the fluorescently labeled T cells, which are kind of immune cells. So that group is specifically interested in under disease model, how the immune cells dynamics change and how the interactions with tissue change. The last example here is another live sample, which is a fruit fly's embryo image. And this group is interested in the development of the embryo. So what they did is they collected the data from the very early stage of the embryo development and tracked the cells divisions and movement along the time and with the 2-photon or multiphoton microscope. So what you may notice here is that for multiphoton microscopy, it is multi-dimensional, which can be 3D, 4D, and also will be multicolor. That's another spectral dimension we add on. So here I want to elaborate more on the process, how an image is collected here, so I can highlight what restrictions we have here with the fluorescence microscopy. So unlike any camera captured images, the fluorescence microscopy we talk about here are mainly called fluorescence laser scanning microscopy, which include a scanning part. And with fluorescence laser scanning microscopy, we rather than shining the entire excitation light on the entire field of view, we have a very small point of laser that scans through pixel by pixel across the XY field of view, 1 pixel by pixel. So you can see that it is a scanning process and the speed can be very slow depending on how fast the scanner moves. And if the laser light hit on a pixel which has a fluorescence in that pixel will emit an emission signal. And if that emission signal is within the one detector's detection band, it will be detected by that specific detector. And we can have multiple detectors on our system with different spectral band. And then the detector will map that pixel onto the displayed image as one single pixel. So that's the entire process of how a fluorescence image is produced on our system. And earlier I mentioned that multiphoton microscopy is unique in that it can image very deep into the tissue, so we can do very thick 3D imaging. So then what it will do is it will move the objective 1 step further down into the tissue and repeat the entire thing, the same thing, and collect another image at a different depth. So with that, we can collect a lot of different depth images and reconstruct it as a 3D image. So that's how we collect 3D image under our fluorescence microscope. So then with that, you may realize there are a few limitations here that can restrict the applications of the fluorescence microscopy. So those are the 3 resolutions of the microscopy that people usually evaluate how it performs. So it has a spatial resolution, temporal resolution, and spectral resolution. Biologists always want every single one of those to be the highest. However, if you increase one of it, the other two ends of the spectrum will decrease at the same time. So those are the three conflicting factors that will be very hard to achieve all of them at a very high level simultaneously, only with our hardware design. So in the microscopy field, there are a lot of fancy designs of different systems trying to achieve highest spatial or highest temporal or spectral resolutions. However, those designs usually will be ultimately limited by the law of physics. So there will be certain points we cannot break the law. So because we are limited by the system hardware design. So we are starting to figure out ways how we can achieve better performance of the microscope with post-processing methods. So then I move to the next part, which will be a project we have done to try to lifting the spectral limitations of the fluorescence microscopy with a very simple and easy to use computational tool. So earlier I mentioned that for fluorescence microscopy, we definitely need to have fluorescence label in the sample. So fluorescent labels, so any fluorophores will have a distribution of their emission signals along the spectrum band. And those here, I plotted 4 very commonly used fluorescent dyes spectrum along the entire wavelength range. And the first thing you may notice that is the emission spectrum can be very broad, and some of them can be overlapping. And also the bar here I plotted, those are our filters that's in front of our detectors, which specify what wavelength band we can detect with a certain detector. And we can see that the detection band are also not very specific, which means it has a range rather than a specific point. And also because it has a range, the amount of detection band we can place here are limited. So with all of this, we can see that the detection signals will ultimately be mixed, even like only with 4 fluorophores here. And also the total number of fluorophores we can detect are also limited. So we cannot detect as many of fluorophores we want. Usually for a very standard multiphoton microscope, people usually detect 2 colors. And for my system, it has 4 detectors. So we usually can do 3 or 4 colors simultaneously. However, you can see here, even with only 3 or 4 colors, we will have what we call channel bleed through issue, which under one detection channel, we can detect multiple signals. On the other side is that biologists always want to see more colors simultaneously, but hardware-wise, we do have the limitations to achieve more colors with the fluorescence microscopy. So there are definitely other hardware designs that can push the limit maybe to add 1 or 2 more fluorophores to be able to be detected. However, those designs can always be very expensive. So then we are trying to figure out if there are ways that we can use post-processing to deal with the channel bleed-through issue we have with the fluorescence microscopy. So there are certain features with the computational tool for what we call spectral unmixing here we want to achieve. First of all, we do want the tool to be blind, which means that we don't want to the tool needs to know some prior knowledge of the spectral information. There are definitely tools on the market that people need to measure the spectrum of all the fluorophores they have in the sample first, and then set into the tool and do a kind of a deconvolution to be able to mix all the fluorophores, which can be expensive and also time consuming and not very realistic. So we do want the tool to be blind. So no prior knowledge is required. Secondly is ideally we want the total number of fluorophores we can like measure or separate be more than the total number of detection channels we have. So the total amount of detection channels can be a hard limit on the detection signals. However, if the tools can separate out more fluorophores based on the current information we have, that will be a better tool for people who want to push the limit of the hardware. Thirdly is co-localization is allowed for the tool, which means two fluorophores can label the same structure and show up in the same pixels. And this kind of labeling is very common with biology studies, and a lot of biologists actually evaluate the co-localization. So the unmixing tool cannot fail when there are co-localization happening in the sample. And lastly is it needs to be computationally light, because this is just the first step for post-processing. We don't want it to be very time-consuming and require a lot of computational efforts. So with those, we first examined all the available spectral unmixing tools and highlighted those commonly used ones on the market. And none of those are having all the features available here. For example, some of them are not blind and would require the prior information about the spectra, and some of them cannot do more fluorophores than the detection channels. And some of the newly developed fancy tools, like someone developed autoencoder for the mixing. It's a little bit overkilling, and it is relatively computationally expensive. So the goal of us is to find something simple to use and is robust and ideally having all the features here we want to achieve. So our solution, we gave a name of Lumos, and I will later on talking about what is Lumos. So when we plot our pixels intensities under different detection channels, intuitively, if the pixels are from different fluorescent, are labeled with different fluorescent dye, they will be distributed on different space in the plot. And so the pixel so the pixels with the same label tends to group together. And then we realized that we can easily use a clustering methods to be able to group those pixels having the similar profile of their intensity under all the detection channels. So the first thing we thought about would be k-means, which is the simplest clustering methods we can use. And the nice thing is that it only requires individual pixels intensity information under each of the detection channel. That's all we need. And we give a name. We call it a learning and supervised mean of spectra, Lumos. And my former lab data analyst gave the name, and inspired by one of the magic spells from Harry Potter, if anyone is Harry Potter fan. And the underlying thing about the Lumos is just k-means, which is very straightforward, and we just initialize with k random centroids and assign the data points to each of the cluster based on the closest mean, and repeat the whole process. And individual spot on the plot here would be every pixel's intensity under the detection channel. Here is a 2 detection channel case. We can have more detection channels. It will be multidimensional. The ultimate goal here is to group pixels with the same spectral signature, which we give the name for the spectral profile of the individual pixels as spectral signature, to group them together so that those pixels can be assigned to a specific fluorophore. One of the drawbacks of k-means, that original algorithm, is that the number of k will be hard to define. However, for this application specifically, the k is already known, and that's the greatest thing here. And the k here, we assign it to be the total number of fluorophores we have inside the sample plus 1. And the additional 1 here is the background noise. So for any microscopy images, there will be by nature some background noise captured. And those background noise would be different, showing differently in profile comparing to those pixels who are labeled with a certain fluorophore. So this would be our K. And K also doesn't need to be equal to the total number of detection channels we have. It can be more than that. And because we have an additional cluster, which is our background noise, the other side benefit from the Lumos algorithm is that we can also do background noise removal with Lumos. So a lot of biologists also like this.
I had a question.
Sure.
Yeah, you talked about a profile, and I guess I'm not quite sure how many, if this was an image, right? How many records, or how many spectral records are you working with here in order to derive your profile? Your profile is based on clusters of pixels, and so can you perhaps you're going to describe that. That seems like that's kind of a key. I guess I was thinking of analogies with, detection in color space, for example, visual.
So you mean like how many different detection channels we have?
Yes, that would be helpful. Yeah.
Yeah. So if we go through the few examples here to show like how well the algorithm perform, you may see that we have different cases of different numbers of detectors we have. I see. Also later on, I have a simulation like results that will elaborate on like how many total number of fluorophores we can have to push the limit. Yeah. I'll get back to you to make sure, this part is answered. Okay. Yeah. So the first example here is the simplest one, and this one example is we started with the same number of fluorophores as the detection channels we have. So this is a fixed cell slide that has 3 colors on it. So we use 3 channels on our system. And what you see here, those 4 images are our original images. And we have 3 colors, the blue, green, and red. And the last one is the combined. And and different compartments inside the cells are labeled with a specific color. And for the blue signal, you may see that it has the similar structure showing up in the green signal as well. So this we can also tell this by looking at the spectral emission spectrum, which is some information available measured by other people online. So we can see that the blue spectrum overlap significantly with the green signal here and can be detected by our green band of the detector here. So that's why we see the double signal in the green channel. So then we applied Lumos on the blue and green channels. And so in this case, we use 2 channels, try to separate 2 colors here. And so we can see that the blue signal is completely separate out from the green signal. And when we plot the spectral signature I mentioned earlier, so we plotted our blue signals signature, the green signals signature, and also the background signature under the 2 detectors here. So we can see that there's distinct differences between the blue signals versus the green signals. So because of these different signatures between those 2 signals, we are able to perform the algorithm on our images to do the separations. And then we moved on to try to push the limit, try to see how about if we have more fluorophores than the channels. Here is one example of a kind of cells that biologists developed. They call it colorful cells because it has many colors in it. And this kind of cell has 3 or 6 different colors labeled with 6 different compartments inside the cell. And we are trying to see if the algorithm will separate all of those 6. And this is our raw image from our system, and you can see that a lot of in between those channels there are a lot of signal overlap, and also the signals are not specific to a biological structure, and the output is very ambiguous. So with this, we sent this image into the Lumos algorithm, and this row here is the unmixed result. So you can see that with Lumos, this algorithm can separate out all the different fluorophores in the sample. And because originally this sample has had 6 colors here, but we only had 5 detectors, then the output will add 1 additional fluorophore here, so that the output has more fluorophores than the original detector numbers. And so this is the ultimate result of the composite, which we can see that all the signals are very specific in each of the compartments of the cell. So this algorithm performed very robustly on those images that has more fluorophores than our detection channels. So the last thing we did was to test the limitations of the algorithm, trying to see under the worst scenario until a certain point, when does the algorithm fail? So we did some simulations under like some theoretical reconstruction of the fluorophores spectrum, and we just add more fluorophores into the simulation, and we assume that we only had 4 detection channels here, and because it's simulating results, we know what's the ground truth here. So we simulated under 3 different scenarios, trying to test the different worst case scenarios here. The first scenario is we adjusted the clustering size, which is a specific structure, labeled structure size. So we assume that when a structure becomes too small, the algorithm will fail to be able to separate that out. That's by nature also some drawbacks of the k-means if there is a imbalance of the structure size. So we can see until a point there is a drop of the performance of the algorithm when the cluster size ratio becomes too small. The second one we tested is we want to see how many fluorophores in total we are able to separate under a 4 detection channel scenario. And under here, we can see that we can separate up around 11 fluorophores here before it dropped down under 0.9, the F1 score. And the last thing we tested here is the signal noise ratio. So sometimes biological images can be very bad in quality, and the signal noise ratio can be bad. So we want to see into how bad the signal noise ratio the algorithm will fail. So it will be around 1/2 before it dropped down below 0.9 for the F1 score. So those are the things we done with our developed tool for spectral mixing, and it turns out to be a very robust, easy to use tool. And when we look back on our table to comparing the features of the desired features, we realize that we can achieve all of the goals here. And actually, a lot of our users starting using the tool and push the limit, hardware limit of my microscope. Some of my users started to be able to see 6 or 7 colors under 2-photon microscope, which has been very rarely achieved before. And also other people in other institutes start using the tools as well and have very good feedback. So that's a small story about one nice, easy way we used to deal with the hardware limitations on our fluorescence microscopy. Before I move on to the next part, I just want to check if we have any other questions. And I don't remember who asked the previous question. Was that Peter? Did I answer your question?
Yes, I think
great.
I think so. I guess I was thinking of the detection channel in your context might be a combination of a detector spectral characteristics as well as an excitation laser. The combination of the two gives you your detection channel. Am I right?
That's a great point. I didn't go very deep into that. So
Okay.
that's a great point. So I didn't touch very deep on that point. So for this example, this is a more complicated example here. I didn't highlight here. We actually were able to detect, like scan twice with two different lasers and detect it under the same detector. That is, we usually call it a sequential scan under our system. You can so in that way, we can actually consider it as what you said, like two detection scenarios here. It is not very easy to do with multiphoton microscopy, and it is mainly because the laser itself is very expensive. So, and most of the two multiphoton microscopy you see on the market would be only one laser. So you at one time you can only use one wavelength. For my system, we have two lasers. So like the maximum you can get excitation would be two different wavelengths. Yeah.
I see.
So,
okay. Very good.
All right. So I'll move to the next part, which is an ongoing project at the lab. We try to tackle another spectrum on our limitations, which are the spatial and temporal resolution limitations of our microscope. So just a revisit of what I showed earlier. So we see that the fluorescence laser scanning microscopy, it scans pixel by pixel, which will make it very slow when we compare it to the camera imaging. Camera imaging would be just like instant snapshot. But for our microscope, I'll show, and for your reference, like a 512 by 512 pixel with our, what I call regular scanner on our system, it takes 1 second. So imagine that if we have many different Z depths, it will take very long to shoot a 3D image. And we can definitely increase the speed with different ways by imaging side. So for example, we can decrease the total amount of pixels we want to scan here. However, there will be a downsampling, and we have the risk to lower the resolution of the image we get. So that's one. And the second one is we can find a faster scanner to do this scanning much faster. And also in the optics field, we do have the faster scanner available. And later on, you may see I name those as resonance scanner. So those scanners comparing to our regular scanners can scan about 30 times faster. However, those faster scanner, because they scan so fast, it will lower the signal to noise ratio we get of those images. So usually those faster scanners images you acquired would be high background noise. On the other side, biologists always want more. They want faster imaging and also high spatial resolution. So faster imaging because they want to do things in life. So they want to capture the dynamics that's happening inside the cell or inside the tissue, but they also want to see the details of the structure in those tissues, so they know what they are looking at. So we can see that hardware-wise, it's hard to achieve both. And so we look into other fields, trying to see what in other fields people are doing, try to boost up the resolution of the image. So current state of the art, there's popular field, image super resolution. People use deep neural networks to map from the low resolution images to the high resolution images. And those were mainly used for camera, or we call natural images. And usually, due to the nature of the camera images, they use the image pairs as the way that they acquire the high resolution images, but downsample it to be the low resolution image pair. and with either bicubic or Gaussian. But in this way, first, it's a mathematical way to downsample it. It may not be true to the real low resolution image. And also there is no noise, usually no noise added, so there is no denoising part with the model. The most popular ones are SR-ResNet or SR-GAN, and it has been very popular back in a few years before. So with this, we were thinking, can we apply the similar model here to the bioimaging field? And so for bioimages, we can see that there are hardware and acquisition time limitations that will limit our image resolution. So we need some computational tools, ideally, to boost up our image resolution to save the acquisition time. So people can acquire image very fast, but with less detail. And because they acquire fast, they will be outputting a low resolution image. So we want to send those low resolution image into our trained network so that it will output a high resolution counterpart to the low resolution image. So the bioimages, our fluorescence microscopy images, have some similarities with natural camera images, but it also have a lot of differences because the optics pathways and how the images are acquired are all different from those camera images. And the unique thing here is you may realize we can adjust the acquisition parameters here easily with the hardware design to be able to acquire both the low resolution image and the high resolution image of the same field of view. So we can easily get the pairs of the training data set for the model. So here is one example just to show you, get an idea how long it takes to acquire image. So this is a 3-color image under my system, and if it's 512 by 512 pixels with our slow scanner, or we call it regular scanner, it takes about 2 seconds here to scan the entire, to get the entire 2D image. If we do it 2048 by 2048, the same scanner, it will be about 1 minute. And it will be impossible for biologists to do it this way. But you may notice that it will give you much more details of the structures here. The last one here is the same 512 by 512 image, but we use a faster scanner here, and then it will increase the speed a lot. Only takes about 780 milliseconds here to acquire the same field of view. But you may notice there are a lot of background noise here and there, and also the structure are not very in detail. So with this, we can see that we can get high resolution image, but it will take time. So for us who developed the tool, we can take the time, try to acquire a lot of training data set with the same field of view and train the model. And then ideally, when biologists acquire the data, they only need to get the last one like this, and then the model can give them a better, higher resolution image that will reveal more detailed structures. So that's the goal here. The approach is what we tried is mainly focused on SR-GAN. We also tried the SR-ResNet as well. I believe all of you are pretty familiar with it. So the GAN network does have a generator and a discriminator, 2 parts, and the loss will be the combine of the 2 parts here. And the generator will transform the low resolution image into the high resolution image, and the discriminator will guide through this process to make sure the output looks like the real high resolution images here. It was developed, the entire network was originally developed for natural camera images, and the network was also trained on the natural camera images. So we want to see if we can directly use the network to our fluorescence microscopy images, and also are we able to train the network to be better suited for the bioimages here. To make it a little bit different from the original network's name, we call it a sRGAN, stand for fluorescence microscopy sRGAN here. Little bit more details about the generator and the discriminator architecture here. It's exactly the same as the original network here. We just used our own images to train the model. The first thing we tested was if we can have a free ride here, just to use the originally trained natural model to directly apply to our images. So, but the end result was not very good as what we can imagine. So left side, this is our low resolution image. Over the right side is our ground truth, which is the high resolution image. In the middle here, it is the output from the originally trained sRGAN model, and we can see a lot of structures are outputted not the way we want. For example, the filament structure here are pretty patched and spotty, and not what we usually see for a bio images. So we think that, we cannot directly use the original network for our bio images. So the next thing we did was to retrain the model. We didn't start with a scratch. We learned the model just by using the originally trained model, add on top with our more training data set from the fluorescence images. We benefit or leverage the original trained network by using the similarities between the camera image and also the fluorescence microscopy images. And add on top, we retrain the network so that we can add additional differences of our images comparing to the camera images. So here are some information about what training data set we use. So we use image pairs here. We have 3 different kinds of data. The first one, we call it the low resolution, which we use the regular scanner acquired 512 by 512. And then we use the high resolution image, which is 4 times more pixel, or 4 by 4 times more pixels here. And the last kind of image here is what we call low resolution, which are images same amount of pixels, 512 by 512 as the low resolution, but we use the faster scanner. So you saw earlier, those images will have higher noise, also a little bit less spatial resolution comparing to the low resolution. So we trained as 2 separate network at current stage. Probably later on we will combine those. So what we train is a low resolution network, which will use low res and high res image as a pair. And we also have a separate network where we use the low res and high resolution image as a pair. So that's what we did. And the training data set type, we have different varieties of biosamples here. And also we want to include different structure types and different labels here to be more inclusive with the model. So we have human kidney, human spleen, and mouse kidney, mouse placenta, and also some synthetic data as well. About 200 patches for each data type. And each patch, so we crop the original image into a smaller field of view. Some of them are 640 by 640. Some of them are 128 by 128. And for validation data set, we use completely different data. Some of them are fixed cells, and some of them are human lung data. For result-wise, we have different ways to evaluate our results. The first most direct way is by viral check. So virally, we want to see if by eye we can see there is a significant increase of the resolution with the output of our network. So what we see here is one closed loop example of different networks output. So over the right here, this is the ground truth, which is the high resolution image. And over the left here is the low resolution image. The top row here is our regular scanners output. The bottom row here is the faster scanners output. So this one has more noise, you can see with the low resolution image here. This column is the bicubic interpolation result. This SR-GAN column is the natural models. It hasn't been trained on our data's output, so you can see it's not very realistic. This one is the SR ResNet output, and this column is the SR-GAN's output. So you can see that the SR-GAN output is closest to the ground truth result. The SR ResNet output is more like a filtered output, or it's more smooth, but it's not very close to the ground truth high resolution image. So virally, the SR-GAN performs much better. We also want to evaluate quantitatively here. So we used several commonly used metrics within the field, like PSNR, the peak signal noise ratio, and the SSIM. Those two metric we want to evaluate the performance quantitatively and compare among different methods here. The last two rows here are our SR-GAN model trained on our data, and also the SR ResNet model, the result. And we can see that those two outperform the other methods we had here. And we also trained the work similarly as what they use for the original SR-GAN is we also downsampled our high resolution data into the low resolution data to use those as a pair rather than using the acquired low resolution image here. So you can see that with that, the training, the model performance was not as good as what we have with the real world data. So one thing you may notice that is, so the bolded ones are the highest performed ones, and the SR ResNet actually outperformed the SR-GAN here, which is actually not surprising by considering what metrics we use here. So this one you can see virally it is more smooth and less noisy, but it doesn't have the details or the similar look as what the ground truth data is. But with those measurements, we cannot have a very good metric to highlight these differences here. So with this, this work is still ongoing, so we're currently seeking other directions here, trying to improve the model. The first thing we want to do is we want to apply it to 3D images here. As you can see, we only focus on 2D image now, and in the future, ideally, we want the model to work in the Z dimension as well, because 3D images are mostly commonly used by the biologists. And also the Z dimension is also very important. Secondly, we want to have other better evaluation metric for super resolved images, specifically for the bioimages here. So as you can see, the metric we currently use are not they are not ideal, not very good to give the results. So we want to see if there are other metric we can use to better evaluate. Third one is we also want to extend to other imaging modalities. Currently, we only focused on the multiphoton images here. We also have the options to use different imaging modalities, or using even the imaging pairs are across the modality. For example, in the imaging field, we also have a type of imaging technique, we call it super resolution imaging, which people developed, which will break the basic physics law of the image resolution you can get. One of the type of the super resolution image is called STAD, and we actually have it at our core facility. So our idea is to collect the image under both confocal and STAD super resolution image, and try to train the model to learn how to go from a confocal image to a STAT super resolution image. Those are just some ideas. And last thing is definitely we want the network to be more general so that it can be applied to many different biology structures. With this, I think that's the end of my talk. And I want to thank my former data analyst, Tristan McRae, who most of the work was done by him. And also the funding was from the University of Rochester. And here is my contact email. If you guys have any questions you want to interact with, feel free to email me. Okay, thank you. And welcome any questions.
I have a question of how long have you been working at these various stages of developing your technique?
How long? You mean?
Yeah, the project.
The first one, the spectral mixing, I think we did it within about 1 year. And those two projects were actually we were running it pretty at the same time. The second project, we probably have been doing it for about 1 year, but my former analyst left, so I had a new person to start pretty soon on the second project. Yeah.
Okay, thanks.
Sure.
I had a question. And that was in the evaluation of the results, and you showed the visual results, and that kind of makes sense. And in your statistical analysis, for example, the similarity measure that you have there, were you using as a reference? Because as I understand that, when I've used that before, as a reference, were you using the high resolution version? I see. Exactly. Okay, I see.
So we didn't spend much time thinking about what evaluation metric we should use. So we directly use the exact same metric people use for like original SRGAN network they developed. They evaluated the similar ways. So we directly use those. It may not be the best for our cases. So we definitely need to find better ways for bioimages here.
Yeah, it makes sense. For example, that's been used, for example, with compressed images, right? Where you have an original and a compressed and an uncompressed. I guess I was also thinking, this isn't really a question, but an observation. It makes sense doing what you were doing because your customers are probably used to looking at the images. I guess I was thinking in some of these it's good to think about the final task. In other words, sometimes I get involved with, I don't know, object recognition or the processing of the images may not be for viewing, it may be for object differentiation, disease state, evaluation, I'm guessing. So it might be an interesting, and I'm assuming that's often done, an interesting way to evaluate it in the context of improvement of those results, because sometimes visually something might not look quite right because it doesn't have the right texture as you saw. But if that texture is really fundamentally random, say photon noise, then that's not key to the task. So anyway, that was my observation, that defining success in terms of the final task might be good.
That's exactly what we thought about, and I'm glad you mentioned about that. And so actually, we had some thoughts about, like you said, the final task. A lot of biologists want to segment a specific structure, so we can evaluate based on the segmented results. Like if the segmented structure, the similar volume or the segmented amount of signals, like how many segmented structures are there, do they match with the high resolution images as evaluation. But it can be, so when we go to the individual cases, it can be like a lot of different ways we can do that, because there are a lot of different tasks. So that's true. I think that's a great point, and we definitely think that that's on our radar. The next things we need to do.
It's also a lot of effort too, because you have to get actual cases to evaluate, right? Okay, thank you.
Oh, thank you for the question.
Any other question?
I only had one other comment, and that was with the k-means or other methods of segmentation, sort of multidimensional data. Is there any application for statistical methods like principal component analysis? Is that something that is used in this field?
Yes, we often use PCA for such kind of analysis. Actually, that's the first thing we tried before we tried the k-means. To my memory, it didn't perform as well as what we wanted to be. So then we changed direction to do it with k-means. Surprisingly, it's performed very well. Yeah, PCA is very commonly used in the field.
I see. Okay, very good. Good to know. Well, thank you.
Yeah, thank you.